Good morning. My name's James Danger. In this video, I'd like to talk about ask the question whether sperm DNA fragmentation is an artifact of in vitro fertilization. Are we spending too much time talking about DNA fragmentation and not about sperm selection? So here we go. One paper I read just recently, after 50 years of IVF, has caused me to think how, in fact, sperm are selected in vitro. There's not a lot of information available, and most of it comes from laboratory and domestic animals. And most of it's been done about 10, 15 years ago and has been out of sight, out of mind, out of sight. But when I read this article in quite a receptive frame of mind, it made me realize that what happens in the female oviduct is actually what we should be doing in IVF. Now, I started IVF, my PhD in 1978, looking at mouse fertilization when there were hardly any publications. And all I did was take a drop of epididymal mouse sperm, added to some eggs, and hey presto, it worked. And then my whole PhD was trying to define that process and see whether I could get to a fertilization of one sperm to one egg. And even when I started IVF, when I finished my PhD, it was still a matter of just dropping a group of sperm which had been selected because of motility in with the eggs and letting the eggs and sperm work it out for themselves. And even when I started doing ICSI in the mid 80s, right the very first wave of ICSI, even selecting sperm out just on motility was the way things have started. And you know what? 30, 40 years later, nothing has changed. We wash sperm with overlay or density gradient or some of the new techniques, and we think they're all motile, we think they're all wonderful, and by and large they are. So it's not really a criticism of it, but that's not what happens in vivo. What happens in vivo, if you read this article, which I strongly recommend, right, that you'll realize that most of the things that happen in human or in vivo fertilization occurs just outside the uterus into the uterotubal junction is where all the selection process occur, where sperm are bound to the epithelium, or the other sperm are removed by a variety of means, and all that's left after intercourse and all the sperm being washed away is this pool of sperm that have been bound to the uterotubal junction epithelium. And only after fertilization, when the egg is in the pure and there's a flood of progesterone running down the fallopian tube, do these sperm then detach, swim up, and fertilize. And if you stop and think, that is actually the process that we should be doing rather than this crazy thing, just picking out any old sperm and injecting it just because they're swimming, because I've done that for 50 years or 40. Right? You drop in sperm with a PVP, you watch them swim around the edge, you pick these fast swimmers out, and in fact, you're doing a very good job, but that's not what happens in vivo. So this talk is about trying to change your mind. I've done quite a few videos now on sperm DNA fragmentation, which is our Achilles heel of IVF. We know sperm increasingly have DNA damage. People spend enormous amounts of academic time processing whether damaged sperm has any impact on IVF. And of course, you put the very best embryo back, and you're never going to show any difference. And so the whole thing has been immensely confusing. And I think it's an artifact of what happens in vitro, and we should change our mind, set to have a very standardized, highly efficient form of sperm selection and not worry about sperm DNA fragmentation. It should not even be on our radar. There are some initial ones like the Zymot technique. Quite frankly, I'm not a great fan of this because the problem here is there's too much seminal plasma carryover, and you still have to wash the sample afterwards. We need to be far more intelligent about how we separate the seminal plasma from the sperm and then the good sperm from the bad sperm. And in fact, the separation techniques probably does need to be a multi-step one, but all in the microfluidic chamber. I think this will be replaced very quickly by more intelligent ones. So it's good to start. The results are a bit better, but I'm not a great fan of this one. This is what's changed my mind. It was this article and a few more. There's quite a few around when you start to dig. In fact, there's a lot. It's just been taken away out of sight. And I think it's time that we should turn our focus, our IVF focus onto this. So there are thousands of articles on IVF. You can see it's keep going up year by year. There's more and more, and you can't possibly manage to keep in touch with it. Here are the ones looking at PGT-A, but this is the one I've started doing now as I do a quick search in PubMed of a keyword and sperm DNA fragmentation. You can see that round about the turn of the century started to become of interest. There's been a steady rise or somewhat plateauing in the last couple of years. 4,500 articles on sperm DNA fragmentation and IVF. Crazy. You'd think we would have solved it by now. I thought I might just pause. I have created on my the YouTube videos a space where a notice board where you can advertise your clinic or your product. And so all you have to do is contact FertAid and we can put your name on the board. And I'm happy to promote your board if you're actively involved in IVF, your clinic, whatever. Just contact me. So, but what if I was to suggest that the issues and concerns about sperm DNA fragmentation were purely and simply an artifact of in vitro culture? I'm not saying it doesn't occur. I'm not saying it's not important in male health. In fact, I think it's really critical in male health. If you're looking at male health, DNA fragmentation is probably the benchmark we should be using to see just what's going on, right? Because everything, DNA fragmentation is purely a symptom of some sort of failure of spermatogenesis. But what if I was to suggest that in vivo, it doesn't matter about sperm DNA fragmentation because these sperm play no role in fertilization and are in all likelihood removed? I'll make a comment. I'm making a claim that oocyte never ever sees a sperm with an elevated DNA fragmentation, even eggs in older women. They just don't see it because the in vivo system removes them. So I like to go back to Louis Baker's paper. I've always known that this is a great paper. Unfortunately, these are mouse eggs, so I don't get too excited and write anything, but I know these are mouse sperm. Human sperm don't have this hook. But the point of the same is, and you know what, just out of interest, this article was almost 20 years old, and you think human reproduction would make some of these freely available. This is, you have to have restricted access. And in terms of information flow, these sort of papers should be made freely available. So the background of the study was to look at the sperm that were binding with to the zona pellucida and look for their DNA. And guess what they found? The only sperm binding to human zona pellucida in vitro were ones that were highly selective for healthy double-stranded DNA. Sperm with a single-stranded or denatured DNA bind less or do not bind at all, probably because it affects defects in motility and more especially morphology. So in a funny sort of way, sperm DNA fragmentation isn't an artifact of IVF, it's an artifact of ICSI. But that problem here is that you put one egg in with millions of sperm and they're all competing and the egg hasn't been built to withstand the assault of a million sperm in one go. It only ever sees half a dozen sperm at best. Now, there's no doubt that the egg does have a lot of fragmentation repair skills. And this also, not 2026, is also an excellent paper. It's really good. Trouble is, it's kind of complicated and you need to be a molecular geneticist to really understand it. But the essence of it is quite straightforward. Findings indicate that the oocyte quality, which diminishes with age, significantly impacts the efficiency of DNA repair process, thereby influencing embryo viability and pregnancy success. While younger oocytes can partially compensate for sperm DNA fragmentation, aging oocytes show reduced repair capacity, contributing to poor reproductive outcomes. These insights emphasize the need for novel reproductive strategies aimed at enhancing oocyte quality and the repair mechanisms. So I know eggs from older women have more aneuploidy, etc., and they form far poorer. However, one can't help but think that if you always use the really healthiest sperm, the ones that were destined in vivo to fertilize, maybe more of these older eggs might just get over the benchmark and actually be able to implant. So just once again, the key thing here is that after ovulation, DNA repair, when the egg has been ovulated, the egg has almost no repair activity. It's been significantly reduced. But once fertilization has occurred, the repair systems are reactivated and sperm, the oocyte initiates repair, employing all these different methods for repairing the DNA. But if the sperm is actually damaged, one would argue that potentially the capacity to reactivate these repair systems becomes damaged in their own right. So if you're using the wrong sperm, the egg really can't the egg can't really reactivate its mechanisms to fix the damaged sperm. It's a chicken before the egg situation. And I don't think I've come across publications talking about the impact of the oocyte to repair, to reactivate its repair mechanisms as a function of DNA damage of sperm. But I bet you there is. So just to finish this little bit, because I just want to, repeating myself, sperm DNA fragmentation is a primary cause of male infertility, affecting ART outcomes and success rates to some extent. The sperm has no DNA mechanism because it's transcriptionally quiet. Its oocyte repair capacity is one of the suggested methods to overcome the situation. However, based on the hypothesis that oocyte age may also have an impact on this talent, these authors looked at to reveal whether the role of female age in repairing sperm DNA damage in this study. And of course, so they compared the outcomes between young and old women with DNA damage, and to no surprise, when maternal age was greater than 40, significantly lower embryo and blastocyst quality, development, pregnancy rates were observed. So this is 2025. Our findings suggest that older eggs may have a reduced ability to repair DNA damage, as demonstrated by poor IVF outcomes with coupled with high sperm DNA fragmentation. Okay. But I want to stop hammering this point because I'm starting to develop after 50 years the philosophy that in fact older eggs don't have to do this. Why make the eggs work so hard trying to fix sperm that they shouldn't be fixed, that they haven't really been designed to fix? So all this stuff with DNA fragmentation is all really interesting. We all love it. There's lots of particle publications. But in vivo, I don't know what happens. All this stuff. But what if I say this in vitro, where the processing of semen to extract motile fraction has been standard practice since the 1980s? But only recently has the focus been on what actually happens in vivo. So I know I discussed before, but I'm returning to us. This is the central part of my theme for this video. What happens in vivo? The selection by the uterotubal junction may extend to chromatin quality. So this is from this article, and these are all linked. There's a really good reference list in this article. Spermatozoa recover from the oviduct, and once again, you have to recognize that when you recover sperm from the oviduct, you normally have to kill the female. That's why we don't do the work in women, right? But in all the laboratory and domestic animal work, they have to kill the animal in some way to find the uterine epithelium of the uterotubal junction, and then they look at sperm. So this is quite a complicated technique and fraught with errors, but the concept is what I'm interested in. Spermatozoa recover from the oviduct exhibit lower DNA fragmentation than those in the uterus. That's fair enough. Together, these findings highlight the role of the uterotubal junction as a multi-level gatekeeper against sperm with structural, functional, and DNA abnormalities. So everything that's bad is actually removed. Beyond the physical barrier, so the sperm have to be able to get there by a variety of tools. This is the key thing. They have to form a reservoir. This is something which I never really fully appreciated in 50 years of IVF. That the sperm, once they get into the uterotubal junction, using a molecular passport, that is certain ligands on the sperm head, are able to bind to the epithelial cells and form a reservoir. They're still alive, they're still swimming, but they're incapacitated. They just sit there, and they sit there for a couple of days. And I think the fact that they just sit there bound to the epithelium waiting for a signal to tell them to detach, I think is a really critical thing. And as I understand it, it isn't until you have a signal from that ovulation has occurred do these sperms start to detach. And it's thought that part of the detachment process is they start to undergo the capacitation process. So once with capacitation, the sperm head has changed, the motility has changed, and that then activates processes to then direct them to the oocyte. But before they're exposed to progesterone or whatever signal about what it is, the sperm remain bound. Now, I think there's a whole area there which the next 10 years, I think is going to be fascinating. And I wouldn't be surprised if, in fact, the way sperm selection is going to go in the past, is that sperm will be prepared 1 or 2 days before the IVF. They will flow over some form of molecular ligand binding to the microfluidic chamber. And then only when a signal is added will those sperm detach. And it's ones that detach, which are the ones you'll use in ICSI. So this is from the article Sperm Navigation in Humans. This is also a fantastic article. Once again, relying on the concept that sperm bind. The process in humans is that separation occurs at the cervix, and the sperm swim up through the uterine medium into the the junction, the base of the isthmus of fallopian tubes, and the sperm are stored there. And it isn't until the oocyte is released and the follicular fluid and other factors are moved down the fallopian tube, and they're calling it progesterone, but there could be a whole lot of other activators. The sperm then detach from the binding and swim up the fallopian tube during a whole lot of processes, mainly to do with chemotaxis, etc. And only those are the ones that fertilize the egg. But after fertilization, the egg is able to release zinc and other project, which actually stops sperm from progressing further. That doesn't happen in vivo. And I think that's one of the problems. I think it may be that, in fact, polyspermy doesn't exist in vivo. I'm sure it does occasionally. But the polyspermy we have in IVF is just an artifact of the sperm being assaulted by millions of sperm, metaphorically speaking, that otherwise has never been prepared to do that. So David Miller is also a tour de force in this argument, and I think his publications are all worth reading. So in this article, Sperm in the mammalian female tract, he makes a point that once again, there is these long uterine horns which vary between women, but at the uterotubal junction, this is where the initial clustering, the reservoir, and the selection of sperm starts to happen. And this is what we need to mimic in our microfluidic chambers. So David makes the point, sperm that reach the site of fertilization are the exceptions and winners of one of the most stringent selection processes in nature. The review, which I recommend, discusses the challenges that sperm encounter and how the few sperm that reach the fertilization site overcome them. The sperm that reach the goal must navigate vasoelectric fluid, swim vigorously and cooperative along the walls of the female tract, avoid the innate immune system, and respond to potential cues as to their direction. Now, I'm going to come back to this immune system because there's something there which I think is really important and may be the key to understand why we don't get more implantations in vitro. So another way to look at improving results relies on the capacity of some sperm to bind to the oviduct sperm reservoir. As stated earlier, only motile sperm with an intact acrosome and the absence of morphological abnormalities, including DNA fragmentation, are capable of binding to the epithelial cells. And then make the point, in vitro models mimicking this interaction include explants, epithelial monolayers, spheroids, and isolated oviduct ligands consistently demonstrate that sperm binding competence correlates with membrane integrity, mitochondrial function, and DNA integrity. This suggests that adhesion is not merely a retention of mechanism for sperm randomly captured by the cilia, but a biomarker of sperm quality. And that, I think, going forward, my friends, is where we need to be focused. Additional studies in cattle report high cleavage rates, etc. But they have a problem in humans because we only ever look at the very best embryos when we put them back, and that masks the importance of going on. The observation that the navigation means essentially selected capacitated sperm cells lead to the idea of one or more of these means to obtain a sperm population enriched with capacitated cells for in vitro fertilization, particularly for ICSI, which is currently the most used artificial fertilizing technique. Now, I'd like to come back to this point and say that, in fact, these articles do suggest that capacitation occurs only on activation with signals from the oocyte. That is normally progesterone, which initiates the capacitation process in sperm, and that capacitation process involves detaching and swimming forward. So it's the detachment from the epithelial cells that's important. Indeed, human mouse bull horse sperm selected by thermotaxis exhibited much higher DNA integrity and lower chromatin compaction compared to non-selected cells. There's some references there. So we're now talking about thermotaxis, which is the detection of heat. I'm going to do a whole video on sperm thermotaxis because you think sperm swim around randomly doing nothing, just trying to find an egg. But in reality, the sperm head is filled with whole sensors. It's like a listing point. It's got sensors everywhere, all cued to signals about temperature gradients and chemical gradients that we have no comprehension, and in vitro don't occur because the in vitro environment is so stale. We need almost, going forward, to create an in vitro environment that starts to think seriously about how it would mimic what happens in vitro. So my comments are, so it seems that in vivo, sperm that are able to bind to the oviductal epithelia are the ones destined to achieve fertilization. And that sperm are characterized by very low DNA fragmentation. In other words, forget DNA fragmentation. If you do your sperm preparation correctly, it's not an issue. You don't have to think. But the key for clinics going forward will be to standardize their sperm selection process. So the only sperm the scientist needs to see for ICSI are the ones which have the capacity to fertilize in the same way as in vivo. Even if in vitro only eggs from older women appear to be affected by elevated sperm DNA fragmentation, it doesn't mean that there is no cost to the zygote. But in vivo, all eggs, regardless of female age, are never fertilized by damaged sperm. Why should we make it hard for oocytes in vitro? I think we've been doing it wrong for 50 years. And of course, there's quite a few articles now. This is almost 7 years ago. There's quite a different ways people have been playing around with of the way that you can select from a sperm population ones that bind. And you know what? I think we're going to use all these different things in the time. There's going to be sperm we end at the end of the chamber are going to be few, but highly competent. But you have to remember that in vivo, the only sperm that swim to the eggs are ones that have just started capacitation. And I think we need to go back and work with our capacitation to keep sperm in a decapacitated state. No, sorry, uncapacitated state until we're actually just about ready to use them. Capacitation works really quickly. This is one which I think has got potential. So, but we have to recognize, start using these words in our vocabulary. Chemotaxis. In this work, it was found that in both cases with selected sperm, there were 3 times more capacitation sperm, which showed less DNA fragmentation and less oxidative stress than semen before the selection. The thermotaxis, we showed in mice and normospermic men that spermatozoa selected by thermotaxis possessed higher DNA integrity compared to seminal samples prior to selection and after selection by swim up. I think we need to go through this process where we need to get rid of the seminal plasma before we start. I think there has to be an initial wash of the seminal plasma, but it's got to go into a media which is not toxic to oocytes but does not allow capacitation to occur. And I suspect it should be something to do with maybe low calcium or whatever. But when the sperm preparation has been made, we have to make sure that when the sperm are free to capacitation, they remain in an uncapacitated state. We don't want any capacitation until just prior to the fertilization. And the authors also showed in mice, I think I've already done that. And I think this is another one. This is a really good article by Michael Eisenbach, who actually shows that thermotaxis is probably considered as a long-range guiding mechanism that guides sperm from the reservoir to the warmer ampulla. So his argument is that the sperm uses different tools at different parts of the process. So it uses thermotaxis to move from the reservoir to the site of fertilization. So sperm can detect immense subtleties in and I think going forward, we should all be using selection techniques that employ thermotaxis. That's what David Eisenbach recommends. Thermotaxis can be considered as a long-range guiding mechanism that guides sperm from the reservoir to the warmer ampulla. Sperm thermotaxis relies on a family of G-membrane protein, couple of proteins that can sense either light or temperature depending on the cell type. And all the 2, 3, 4, 5, they're all present in human and mouse sperm, but in their eyes, opsin 2 and opsin 4 were shown to be crucial for sperm to perform thermotaxis, each mediating different signal. It's almost as if the sperm have had all these techniques available and we just haven't seen them. I think we need to open our eyes up. The sperm just don't swim randomly. They are looking for signals of where to swim. And once again, I think we're going to the new techniques for doing sperm selection. I don't think we can get away from it, but they could look at motility, chemotaxis, thermotaxis, and rheotaxis, and it may well be, excuse me, that you'll take sperm, wash them, run them through maybe a motility gradient, and then keep them in storage. And then from the storage, you put them into a second microfluidic chamber that maybe does thermotaxis. And right at the very end, you add in a chemotaxis like progesterone. And so I can imagine that sperm will be processed in multi levels. And it won't just be like using AI to pick out a sperm which they think is really good. I think the problem with AI selection of sperm, even though it's really clever, right, they're actually picking out the wrong sperm. And I'd just like to finish off by saying, so my argument is that all IVF and ICSI cycles should use sperm preparation methodology that only renders sperm with ultra-low sperm DNA fragmentation, because that's what happens in vivo. It's just because when transferring the very best embryos, this masks any effect of sperm quality and does not mean there is no impact. Good practice demands application of the best technology. And in reality, we know very little about the impacts of disturbed fertilisation, even on healthy young oocytes. And if you think we know everything, just look at your pregnancy rates and you realise that you put back your best euploid embryo, the best blastocyst with autism. In some cases, you might get close to 60%. But if you stand back and look, it's more like about 35, 40%. You've been seduced in some way by looking at the very best euploid and thinking that's actually what's happened. But in fact, a lot of eggs are poor, and maybe they're poor because we haven't recognized that they've been damaged at the start. So there's a whole lot of work now being done about fertilization and proteomics in the oocyte, and how that, on day one of fertilization, is affecting the cells allocated to the inner cell mass and the overlying trophectoderm. So actually, I think we know very little about the impacts of disturbed fertilization on even healthy young oocytes, and it's time that the IVF industry moved to upgrade and standardize the sperm selection process. And I don't think Zymot is the one. I think it needs to be the new form. And just to prove my point, I have already done a video on this. You should hunt around and find it. It's a work from some Moroccan authors, looked at phospholipase C. And phospholipase C is the enzyme in the sperm head that once it gets access to the female cytoplasm, causes the release of waves of calcium oscillation, which are the first chemical signal of fertilization. And everyone has ignored that in the past. But in fact, I remember going to a talk about 20 years ago where they first started to show this, and I was flabbergasted to show the changes in this, the waves of oscillation can impact the rate at which embryos will develop 3 or 4 days later in the mouse, and I'm sure also in the human. But get this is the point I want to make is, in the fertile men, they have a lot less phospholipase C, but more DNA fragmentation. So just because the sperm got DNA fragmentation, and you think the DNA can be repaired by the egg, in fact, if they have damaged DNA, they have damaged calcium oscillations, which will damage the reactivation of the pre-peg mechanisms. It's wrong to ask the egg to fix the damage. They shouldn't have to do that. It's got more important work to do than fixing damaged DNA. So just because younger eggs may be able to accommodate damaged sperm DNA and repair the problems, and older eggs may struggle, why make any egg work harder than necessary? The quiet embryo hypothesis states that the less energy used, the more internal energies that the less energy and the more internal stores are used, the more delay in development will occur. That is, my point is, in vivo, theoretically, sperm with damaged DNA do not get to fertilize an oocyte ever. In vivo, the egg does not have to expend time or extra energy to fix really damaged DNA. It's just minor damage, I assume. So now that we're just starting to have the tools to improve sperm selection, we should universally apply these to IVF and especially ICSI. So once again, my name's James Danger. I've been more than 50 years in IVF. Not a medical person. This information is not medical advice, but it may help you know what's going on. If you like what I'm doing, press subscribe and tell everyone else about my work. Its aim is for education and to improve IVF. Now that I'm voluntarily retired, my aim is only to educate. But I do have an IVF channel at fertile.com that does online EQA and training, and you're welcome to explore that. So at this point in time, I'd like to thank you if you've managed this far. I know IVF is a personal journey that involves the actions of many people, techniques, and ideas, and it helps to understand all aspects of that journey. At this point in time, I'd like to say thank you for your time and goodbye.